The next type of biomolecules we have are nucleic acids. See, we know the characteristics of a species are transferred to its next generations in many ways. Most of the characters are transferred to the next generations. This transferring of characters from one generation to the other generation, or transferring of the traits or the information from one generation to the other generation, is called heredity. Now, the biomolecules which are responsible for the transferring of this information from one generation to the other generations are basically chromosomes which are present in the nucleus of these cells. Now, each cell is having these chromosomes, which are a set of chromosomes, which basically are made up of what? Proteins and other type of biomolecules which are called nucleic acids. So, nucleic acids are main hereditary material responsible for the what? The passing of the genetic information from one generation to the other generation, passing of the traits or other characteristic features from one generation to the other generations. Now, chemically they are polynucleotides. That means they are polymers, and they are polymers of the monomers called nucleotides. Now, these nucleic acids are of two types, DNA and RNA. DNA is called deoxyribonucleic acid, and RNAs are ribonucleic acids. Chemical composition of the nucleic acids shows that, which we can easily get on hydrolysis of any one of these. It shows that these nucleic acids are made up of three types of molecular species. One are pentose sugar, 5 carbon containing sugar molecule, carbohydrates, phosphoric acid molecular species, and nitrogenous bases. These are heterocyclic nitrogen containing molecular species which are called nitrogenous bases. Now, we have to understand the composition of the nucleic acids. The pentose sugar which these nucleic acids consist of are having, they are mainly of two types. They are either R-ribose or deoxyribose, or we call it which is there in the DNA. This deoxyribose, as the name indicates, is deficient of one oxygen. That's why it's called beta-deoxy, D2-deoxyribose, and it's called beta-D-ribose. Now, these pentose sugar, how they differ from each other, you can see here, this is the first carbon atom and this is the second carbon atom. This is the first, this is the second. In deoxyribose sugar, the second carbon is not having any OH group, whereas the ribose sugar is having, yes, even one OH group on the second carbon atom also. So, that is how we say that the pentose sugar which are there in these nucleic acids are of two types. One is ribose, another is deoxyribose. Now, deoxyribose is only present in DNA, whereas ribose is only present in RNA. So, that's the basic difference between the DNA and RNA. Now, let's talk about nitrogenous bases. Nitrogenous bases are nitrogen containing heterocyclic aromatic compounds, which are actually of 5 types. They are called adenine, given as symbol A, guanine called symbol G, cytosine given symbol C, thymine given symbol T. There is one more which is called uracil U. Now, how they are distributed in DNA and RNA. Remember, these 4 are in DNA, like adenine, guanine, cytosine and thymine are in DNA. Uracil is never present in DNA, whereas always remember, the uracil is only present in RNA and there is no thymine in RNA, whereas uracil along with adenine, guanine, cytosine is in RNA. So, this shows that a nucleic acid, whether DNA or RNA, at any time is containing only 4 types of nitrogenous bases. It is A, G, C, T for DNA, whereas A, G, C and U for RNA. Exclusively uracil is in RNA, exclusively thymine is in DNA. That's again is a major basic difference, structural difference between DNA and RNA. And all these units are linked to each other through phosphoric acid molecular species, and let us see now in the structure of the nucleic acids.